It just almost makes it feel worse. As long as it's short. Yeah.
You're so James says that he'll volunteer to run for the lab tomorrow if you talk about it.
Oh, okay.
Is he not coming to class? Is he not coming to class?
I don't know. Let's do it.
Just here 5 minutes.
He's got packages waiting on first step, and he's afraid that his packages
It's stolen.
is something like that. Yeah, fine.
No, actually.
Thursday.
Very well.
Honestly, we did for the first like 15 minutes, we just reviewed the before. How many slides do you think we got to this system? Like 4? Like 4. Yeah. It's all a pretty long story.
Well, where's that? Berg Tim. He's expecting.
He talked about his crackhead friend who started an electric car. He's now making more than any of us will.
Nice to you guys. Welcome back.
I wish.
I really was out there.
How he was a drug addict? Yeah.
Yeah, honestly.
Thank you. Oh, perfect. Thank you.
These are my exam.
I should go to the library. I was like,
I tried with one piece. One, and it's worse than with multiple. Stupid. Yeah. I'm about to just grab one.
Bracken, can you hear me all right?
Yes, I can.
Okay. All right. Good afternoon, all. Welcome back. Hope your spring break was everything you hoped it was and more. I spent my spring break working, grading, editing, writing. I got a lot done. But yeah, it was busy. But hey, that's okay. It keeps me out of trouble. Okay, Bracken, did the slides move?
that could be the things on that stuff.
The thing's all messed up? Oh. The slides move, Bracken?
Yep, they're moving for me.
They're moving? Okay, great. Okay. Okay. Now, before we get started, we have a little bit of housekeeping. to deal with. For tomorrow, we're going to do a endocrine study looking at the efficacy of insulin. Now, it's well known that insulin's efficacy is greatly influenced by exercise. So we are going to have 3 groups. We're going to have the moderate exercise, light exercise, and couch potato group. So for those of you in my section tomorrow, please raise your hand. Okay, I need people who will do moderate exercise like jogging. Anybody want to run for me tomorrow? Ben, you guys, Nathan, Ben, you want to run? Okay, so come ready to run. Okay, so after you drink the glue, you guys have to come back though for the glucose readings. Okay, don't go out, la, 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 we're out in Concord. Okay, no. Make sure you're back. Set your watches 30 minutes. You have to be back by 30 minutes to get your glucose read. Okay, 2 guys to run. Who wants to walk? Okay, Jamie, and anyone else want to walk? Okay, Bethany will walk. Okay, all right, 3 walkers. So you guys, you'll walk. You can walk wherever. You walk the track. Doesn't matter to me. Just be back within a half an hour for a glucose reading. Okay, the rest of you are going to be couch potatoes. All right. So bring some homework to do. Watch a video, whatever. You're going to work on your phone, whatever. You'll be couch potatoes. Okay, so have the walkers, the runners, and the couch potatoes. And we will look at how physical activity will influence your insulin and how it will bring your glucose down after. You're going to drink glucola. It's 25% glucose. You'll drink 4 mils per kilogram body weight. So please calculate your body weight in kilograms. Take pounds, divide by 2.2. That will give you kilograms. And then you'll drink, as we said, 4 per kilogram. So come please with the amount you'll drink calculated. The glucola is disgusting. It's horribly sweet, but you only have to drink it once. And before you drink it, I will put some lemon juice in it, so it'll taste more like lemonade. It's still gonna be really sweet, so just warning you, okay? If really sweet stuff makes you sick, please tell me so that you can drink it slowly. Or if you're one of these people that's reactive hypoglycemia, and if you drink it, you'll just go into a fit or something, then you can be exempted from drinking it. But the rest of you, without that, Caitlin, why are you looking at me as if I'm putting you through the ringer or something? It's just glucola. It's just glucose. It'll be fine. You'll be all right. All right, and then for Thursday, we'll sort out who wants to run, walk, and be a couch potato for Thursday. Okay. All right. And Bracken, hopefully you'll be back. So by Thursday, you'll be able to be assigned to a group by then. Very good. So we're going to talk about the endocrine system. The endocrine system is a regulatory system. And the endocrine system consists of glands. They're endocrine glands, so they are ductless. They secrete their materials, their hormones, straight into the bloodstream. That's why they're endocrine. The target organ is usually far away from where the gland secretes its hormone. That's the hallmark of endocrine signaling. The tissues that receive the signal and respond to it are usually far away from it. Some of these hormones are secreted by neurons. They might even be neurotransmitters. So those are neurohormones. Neurohormones won't work as neurotransmitters. Instead, they'll be secreted into the bloodstream. Instead of them being released in a synapse, they're going to respond differently in this case. Hormones will affect targets. They're actually going to affect the gene expression pattern of those targets. Signaling that starts at the cell membrane will result in changes in gene expression. So here's a table from chapter 11 of your book, shamelessly stolen from your book. This is a very good table that summarizes some of the hormones and neurohormones as well, and the organs, the glands that secrete them, and their function. So I would highly suggest you spend some time with this and get some ideas. Some of these we will not talk about in this chapter. Leptin we'll talk more about when we get to metabolism. Gastrin we'll talk more about when we get to the digestive system. Same thing with testosterone and vitamin D. We'll talk about those later. Most of these others, we already talked about melatonin. We'll talk about estrogen and progesterone when we talk about reproductive system. And erythropoietin, we'll talk about when we get to the circulatory system. The rest of these, well, secretin, cholecystokinin, that's digestive as well. The rest of these we'll probably talk about in this chapter. So structurally, there are 4 main types of hormones. One of these are proteins or polypeptide chains. They're either small polypeptides or they're larger polypeptides. Both polypeptides and proteins that have carbohydrates attached to them, glycoproteins, those cannot cross the membrane. They have to bind to a receptor embedded in the membrane. Amines are derivatives of amino acids. Either the amino acid tyrosine or in some cases, the amino acid tryptophan. And those small molecules are called bioactive amines. They too are very polar. They cannot cross membranes. They bind to receptors embedded in the membrane. Steroids are quite hydrophobic. They can charge right through the membrane, and they're going to bind to a target inside the cell. Inside the cell. The bioactive amines, they're derived from those 2 amino acids, and they're secreted by the adrenal medulla, the thyroid, and the pineal gland. Pineal gland makes melatonin. That's a derivative of tryptophan. The adrenal medulla releases epinephrine and norepinephrine. Those are derivatives of tyrosine. And the thyroid releases thyroxine or triiodothyronine. Those are both derivatives of tyrosine. Polypeptide and glycoprotein hormones range from the very small antidiuretic hormone, oxytocin, they're both quite small, 8 amino acids, to glycoproteins like follicle stimulating hormone, luteinizing hormone, or thyroid stimulating hormone. Parathyroid hormone is 84 amino acids, but it's in length, but it's not glycosylated. It's not a glycoprotein. So these are your peptide hormones. These here are your glycoprotein hormones. Again, these cannot cross the membrane. They have to bind to a receptor embedded in the membrane. Steroids, all steroid hormones, all of them are derived from cholesterol. Every one of them. That cholesterol can come from your diet or can be made in your body. And it's delivered to tissues by lipoprotein particles made by the liver. So this is another picture stolen from your book. Cholesterol, as you see, is also the precursor to make progesterone. You see, it's just 2 steps from cholesterol to progesterone. You can then take progesterone and use it to make cortisol. Progesterone is also used to make androstenedione. Androstenedione is used to make testosterone. And then testosterone is used to make one of the major estrogens. There's three main types of estrogens. One of the main ones is estradiol. And that is what's made in the follicles. And all this started up there with cholesterol. These are the hormones made by the thyroid. Thyroxine, otherwise known as tetraiodothyronine. So you can see the tyrosine here. Chemically, you add the iodine and you link it to another tyrosine. Add the iodine, 4 of them, that's thyroxine. Take one of the iodines off, you now have triiodothyronine or T3. T3 and T4, that's what is detected when you get a thyroid hormone level test. Typically, to test your thyroid, the physician will order a thyroid stimulating hormone test because that test is easier and cheaper. The T4 and T3 test is more expensive and is a tad more difficult and is used if there's a problem with TSH, if your TSH is too high. All right, let's take a look at how steroid hormones do their business. Okay, let me make sure the sound is all right for this. Give this a shot, see if this works.
Steroid hormones are not water soluble. They travel in the blood attached to protein carriers. When steroid hormones arrive at their target cell, they dissociate from their protein carriers and pass through the plasma membrane of the cell. Some steroid hormones bind to specific receptor proteins in the cytoplasm and then move as a hormone receptor complex into the nucleus. Other steroids travel directly into the nucleus before encountering their receptor protein, not shown. The hormone receptor protein, activated by binding to the hormone, is now able to bind to specific regions of the DNA. These DNA regions are known as the hormone response elements. The binding of the hormone receptor complex has a direct effect on the level of transcription at that site. Messenger RNA, mRNA, is produced, which then codes for the synthesis of specific proteins.
Now, based on the way steroids work, would you expect steroids to work slowly or quickly? They work quickly. Oh, there's that buzz again. There. They work quickly. Because they don't need to mess with receptors here and all this other signal transduction, this, that, and the other thing. They go straight to the DNA source and they activate gene expression. So steroids tend to work quickly. They go straight through the cell membrane. They don't need a receptor embedded in the membrane. They address targets inside the cell. All right, let's now take a look at a particular hormone, aldosterone. You know what, let's not look at that one. Let's look at one. We'll look at this other stuff. Well, let's do this one. Okay, here's another bit about how, let's get it so that Bracken can actually see this. Okay.
or intracellular mediators affect changes inside of cells when a
Let's try that again. Okay.
second messengers, or intracellular mediators, affect changes inside of cells when a signal molecule binds to a receptor on the surface of the cell's plasma membrane. The binding of the signal molecule, the first messenger, to its receptor activates a nearby G protein. Depending on the target cell, the effect of the G protein varies, typically activating one or two different sites. Some kinds of messenger systems involve activating the production of cyclic AMP. When a signal molecule, such as the hormone epinephrine, binds to a cell surface receptor, it activates the G protein on the inside of the cell membrane. The G protein then stimulates adenylate cyclase to produce large amounts of cyclic AMP within the cell. The cyclic AMP then binds to and activates a target protein such as alpha kinase, which adds phosphate to specific proteins in the cell. The effect of activating cyclic AMP is a cascade, an amplification, as each molecule of cyclic AMP activates many enzyme molecules, each of which activates many more molecules. Multiple copies of activated alpha kinase have phosphorylated even more molecules of some specific protein. The effect of this phosphorylation depends on the identity of the cell and the proteins that are phosphorylated. Calcium ions are involved in other second messenger systems. In response to a signal molecule from other cells, the cell surface receptor activates, which in this case activates the enzyme phosphorylation. This enzyme catalyzes the production of inositol triphosphate, or IP3, which diffuses through the cytoplasm to the endoplasmic reticulum. The IP3 binds to and opens the calcium channel, allowing calcium ions to flow from the endoplasmic reticulum into the cytoplasm. The calcium ions trigger proteins sensitive to calcium to initiate a variety of activities. Whether triggering phosphorylation or the release of calcium ions, second messenger systems or intracellular mediators allow signal molecules attaching to the outside of the cell to have an effect on activity inside the cell.
So that shows you what happens once you get the hormone binding to its receptor. You have this machinery inside the cell that is put into motion. And you get a response essentially from that cell. Usually you get some sort of change in gene expression. And that change in gene expression all began with an event at the cell membrane. Okay, come on. Okay, there we go. Now, some hormones are made as precursors. Those precursors, either some polypeptides need to be clipped from a larger precursor to their active form. Those inactive forms are called prohormones. They only become active after some sort of processing. And here's some examples. So vitamin D3, which you may get from a supplement or you might get from your diet. It needs to undergo two reactions, hydroxylation reactions, one of which is catalyzed by an enzyme in the liver, another of which is catalyzed by an enzyme in the kidney. And you make 1,25-dihydroxy vitamin D3. That's the active product. So the vitamin D3 that you take is a prohormone, and it takes about 2 weeks actually to completely activate it. Testosterone is in many ways a prohormone. It will need to be converted to DHT, dihydrotestosterone, or in some cases, testosterone is converted into estradiol 17 beta. Or T4, thyroxine, you have to remove one of those iodines And now you get the active form, triiodothyronine, T3. That's the active form. So these glycoprotein, polypeptide, bioactive amine, hormones, they have to bind to a receptor. When they bind to a receptor, you initiate this response inside the cell. You make a second messenger, and it culminates essentially in changes that will cause changes in gene expression to the target cell. Steroid hormones, because they're insoluble, your blood doesn't just throw not very soluble things into the blood. It has a carrier. Steroid hormones will usually bind to carrier proteins. That carrier protein will be in an equilibrium with free hormone and the bound hormone. Only the free hormone can enter cells. The bound hormone cannot. Those carrier globulins are usually made by the liver. So if your liver is not very functional, you're going to have more free hormone around, something to consider. What happens when one tissue receives signals from two different hormones? Those effects can be synergistic sometimes. Those synergistic effects can be additive. You get this much effect with one, with two, you get twice as much, or it just gives you more of the same. Or they can be complementary. So a good example is norepinephrine and epinephrine, their activity on the heart. That's additive. You get increased heart rate, you get increased force of contraction. That's additive. The activity of follicle stimulating hormone and testosterone on sperm development, however, is complementary. Follicle stimulating hormone is required to initiate spermatogenesis. But to maintain it and complete it, you need testosterone. So one starts, the other finishes. So this one's the starter, this one's the bullpen, and is the closer. How about the effect of hormones on memory tissue? If estrogen, cortisol, prolactin, oxytocin, estrogen is involved with proliferation of tissue in the mammary gland. Cortisol and progesterone are involved with differentiation of that tissue into lactiferous ducts and lactogenic tissue. Prolactin takes that differentiated tissue and tells it to make milk protein. Oxytocin causes the myoepithelial cells to contract and drive the milk from the glands through the lactiferous ducts into the nipple for the baby to receive. Notice lots of back scratching going on here. Those are examples of complementary action. Then you have this thing called a permissive effect, a permissive effect. One hormone has to act on a tissue in order for it to respond to a different hormone. And the best example here is the uterus. Estrogen actually causes the uterus to proliferate, and then it causes those cells to make the receptor for progesterone. Without estrogen, the uterus would not respond to progesterone. Progesterone then causes differentiation into the glycogen glands that you find in the secretory glands that you find in the endometrium. That's a permissive effect. Hormones can antagonize each other as well. Insulin and glucagon, good example. Insulin lowers blood sugar. Glucagon raises blood sugar. Insulin promotes fat formation. Glucagon promotes fat breakdown. Insulin promotes glycogen deposition in the liver. Glucagon promotes glycogen degradation in the liver. Good example of antagonistic activity. All right, let's go ahead and take a look at some of the individual glands. The pituitary gland. The pituitary gland has two different lobes. Those lobes have two different functions and they develop from two different tissues. So the anterior pituitary develops actually from a downgrowth, an upgrowth of the roof of the mouth called Rathke's pouch. So you get the pharynx, that's endodermal tissue. You get this little pinch off here. It pinches off and then it fuses with a downgrowth from the hypothalamus. That downgrowth becomes the posterior pituitary. This pinching off from the roof of the mouth fuses with it, that becomes the anterior pituitary. Consequently, the anterior pituitary, if you take a section of it, very glandular looking. Posterior pituitary, when you take a section of it, is very neural looking. So here's a picture from your book. Here's your anterior lobe, sometimes called the pars distalis. Posterior lobe, you have a pars tuberalis. It's like a seam in between the two. There's a stalk that connects it to the hypothalamus called the infundibulum. And then the optic chiasm is right above the pituitary. It's a nice little landmark to tell you where you are in the brain. The anterior pituitary secretes trophic hormones. Trophic hormones. They're called trophic hormones because they cause the growth of the organ. If you withdraw the trophic hormone, that organ will atrophy. If you oversecrete that trophic hormone, the organ will overgrow. And it will get too big. It undergoes hypertrophy. So all of these trophic hormones released by the pituitary, anterior pituitary, will have this suffix tropin at the end. You see tropin, they are a hormone secreted by the anterior pituitary. Here is a nice list of those trophic hormones. So ACTH is adrenocorticotropic hormone. Its target is the adrenal cortex, not the medulla, the cortex, the adrenal cortex. It's a peptide hormone. Thyroid stimulating hormone, TSH. That's also called thyrotropin. Its target is the thyroid gland. It's a glycoprotein. GH, otherwise known as somatotropin, growth hormone. It targets kind of everything, but the big ones are the skeleton and the muscles. Then follicle stimulating hormone, which is a gonadotropin. And this partner in crime, luteinizing hormone, another gonadotropin. Their targets are the gonads in both males and females. And then prolactin, which doesn't end in tropin. But its target, of course, is the mammary glands and then other accessory organs as well. The secretion of these hormones is regulated by the hypothalamus. The hypothalamus releases hormones that either activate the release of trophic hormones or inhibit the release of trophic hormones. Let's take a look at these particular hormones that are involved with regulating the release of these trophic hormones. So the first one over here is thyrotropin releasing hormone. This will cause the release of thyroid stimulating hormone. Then we have corticotropin releasing hormone. Otherwise known as CRH. That will cause the release of, which hormone do you think? Yeah. Adrenocorticotropic hormone. Then you have another one called, and this is gonadotropin releasing hormone. Ah. My G's look like they're on drugs. That causes the release of follicle stimulating hormone and luteinizing hormone. And then you have growth hormone releasing hormone. And there's no real mystery as to what this one will cause you to release. That will cause a release of growth hormone. All right, what about the inhibitory ones? There aren't very many inhibitory ones, as it turns out, because these hormones, when they rise in their levels, they will tend to feedback inhibit the release of the hypothalamic release of these releasing hormones. That's why you don't need a lot of inhibitory hormones. One is growth hormone inhibitory release hormone. There's a much nicer name for this hormone, somatostatin. Now, somatostatin is also made by the GI tract as well. It's made in other tissues as well, not just in the brain. Then there is a so-called prolactin inhibiting factor. And this turns out to be dopamine. And in fact, there are certain drugs that are given for schizophrenia. They inhibit dopamine receptors. And one of the side effects of them is increased prolactin production. Increased prolactin production in males decreases sperm counts, and it can cause subfertility. And in women, it of course can cause illegitimate lactation, as it's called, and that of course can be remarkably embarrassing. Now, as it turns out, the hypothalamus and the anterior pituitary have this blood system that runs from the hypothalamus straight to the anterior pituitary. This is called the hypothalamic hypophyseal portal system. The hypothalamic hypophyseal portal system means that when those releasing hormones are released into the blood, their first stop is the anterior pituitary. So this releasing hormone, trophic hormone release is very, very, very fast. And you can see the targets of the different trophic hormones here. Growth hormone, prolactin, thyroid stimulating hormone, adrenocorticotropin, and the gonadotropins, follicle stimulating hormone, and luteinizing hormone. What happens if you don't make enough growth hormone early in life? You suffer from hypopituitary dwarfism. Now, hypopituitary dwarfism can be treated with injections now of human growth hormone. Why? Because human growth hormone gene was one of the first genes that was cloned by the Genentech Corporation. Can you guess how growth hormone was extracted before it was cloned? Extracted from cadavers. It took 42 cadavers to get enough material to treat somebody for 1 year. Imagine how expensive that was. Then you have to get people to volunteer to have their brains ground up, essentially, to be able to extract the growth hormone. So the cloning of this product has brought the price way down, and now has made it available for people with hypopituitary dwarfism. Now, growth hormone levels are much lower in adulthood. If you don't secrete proper growth hormone during adulthood, you can have something known as pituitary cachexia, which also is known as Simmons disease, where you have a general wasting of bone, muscle, and fat tissues. Now, if you make too much growth hormone, you can have a condition called gigantism. And excessive growth hormone production during adulthood can, after you stop growing, essentially, you'll get massive growth in the soft tissues. The jaw will start to grow forward. You have a condition called acromegaly. Are any of you fans of the old Addams Family show? Remember the gentleman who played Lurch in that show? He had acromegaly. I can't remember the actor's name. He also did a lot of voice acting. He had this remarkably deep, rich, beautiful voice. And he was in Star Trek. He's in a few episodes of Star Trek. That's an example of a chap with acromegaly. So Lurch, if you ever watched the old shows with the butler Lurch, he was almost 7 feet tall, as I recall. He was 6'11" or something like that. And as he got older, you could start to see the jaw start to jut forward. Man, I can't remember that guy's name. Anyway, that's an example essentially of someone who had acromegaly. What about the posterior pituitary? The posterior pituitary is largely composed of axon termini. Those axons extend from neurons whose cell bodies are in the paraventricular and preoptic nuclei in the hypothalamus. And they release either antidiuretic hormone, sometimes called vasopressin, and oxytocin. Antidiuretic hormone, if you have high blood osmolality or low blood volume, you'll release this and it will help retain water from the urine. It will also cause vasoconstriction of blood vessels, at least at high concentrations. That will decrease osmolality and increase blood pressure. Oxytocin dilates the cervix and stimulates uterine contraction during childbirth. It stimulates myoepithelial cells in the breast tissue to eject milk during breastfeeding as well. That's called the letdown reflex. And aside from motherhood, its levels are generally pretty low, but they increase after pleasant social interactions. So hugs and physical contact and out of boys, out of girls, and so on and so forth. And they are responsible for the afterglow after sexual intercourse as well. Here's a nice picture from your book that shows you the supraoptic, I said preoptic, I'm sorry, supraoptic and paraventricular nuclei with their cell bodies in those areas, the long axons that go through the infundibulum. That's called the hypothalamo-hypophyseal tract. You then have their axon termini right over those blood vessels. They are stored in little vesicles. Those little vesicles are known as herring bodies. Herring bodies. And when they receive the signal, they will release their peptide hormone straight into the blood. The release of these hormones is controlled by neurological and endocrine types of events, particularly oxytocin. So it's the mechanical suckling, or in actually some cases, mothers will actually hear their baby cry and they'll start to lactate and start to release milk, or just seeing your baby sometimes can do that too. Those are examples of neuroendocrine reflexes. Neuroendocrine reflexes. With the case of ADH, it's osmoreceptors that are in the hypothalamus. They detect the rise in osmotic pressure of the blood. Its secretion, however, is inhibited. Once the blood volume goes up, you have stretch receptors in the left atrium. That will actually prevent production, stop production of ADH. So that's another good example of a neuroendocrine reflex. Regulation of the anterior pituitary, as we said, is by hormonal rather than neurological means. So you have the production of the releasing and inhibiting peptides. They're released into the blood. In particular, they're released into a portion called the median eminence. And the median eminence has these capillaries that get drained by venules in the stalk of the pituitary, as you see here. So there's the median eminence, as you see there. And here are the releasing hormones. They're released into these vessels here. And the drainage goes straight to the anterior pituitary. That's your hypothalamo-hypophyseal portal system. And this gives you an idea essentially of the size of some of these hormones. TRH, as you can see, is tiny, 3 amino acids long, very tiny. Gonadotropin releasing hormone, 10 amino acids, pretty small. The winner here is growth hormone releasing hormone at 44 amino acids. That's still not very big as proteins go. Most of those are relatively small peptides. So the anterior pituitary is also regulated by feedback inhibition by the target organ. So the secretion of adrenocorticotropic hormone, if your corticosteroid levels increase, you'll get feedback inhibition on the release of ACTH. Thyroid stimulating hormone is also negatively regulated by a rise in thyroxine. So negative feedback is going to regulate the release of tropic hormones by the anterior pituitary as well. This has been observed for a long time. So in animal science, if you castrate, for example, a sheep, which is done relatively often because usually you take the strongest sheep, you let that sheep reproduce. The others, you gild them, they're called wethers, essentially. Castration causes a rise in gonadotropins because the testosterone is not there to shut down production of gonadotropins. If you remove the adrenal glands, ACTH will now rise. If you remove the thyroid stimulating hormone will now increase. Thyroxine not only inhibits thyroid stimulating hormone secretion, it also inhibits thyroid thyrotropin releasing hormone by the hypothalamus. So that particular hormone inhibits at two places. So the hypothalamus releases thyrotropin releasing hormone. It tells specific cells in the anterior pituitary, the thyrotropes, to release thyroid stimulating hormone. When thyroxine levels get high enough, they will feedback inhibit the thyrotropes in the anterior pituitary so that they're not as responsive to thyroid stimulating hormone. And then when it comes to the gonadotropins, their inhibition, as you see here, can go straight to the source. They will actually inhibit the hypothalamus from releasing gonadotropin releasing hormone. And they will decrease the responsiveness of the gonadotropes that release gonadotropin in the anterior pituitary from responding to gonadotropin releasing hormone. So you have decreased amounts of the releasing hormone, gonadotropin releasing hormone, and you have just decreased responsiveness to it. That's going to shut down production of sex steroids in that case. Now, what happens if estradiol levels get really high? In that case, you can get a positive feedback system where you get this pulsatile release, essentially, of gonadotropin releasing hormone. That causes a spike in follicle stimulating hormone and releasing hormone. That's responsible for ovulation. We'll cover that more when we get into the reproductive system. Any questions about the pituitary? Pituitary is easily the most complex of the hormones. Very good. Let's go to the adrenal glands. The adrenal glands sit right on top of the kidney. When you did your CATs in 263, the adrenal glands are more towards the midline. They're not on the kidney. They're like next to it, medial to it. It's different in a cat than in a human. In a human, the adrenal gland looks like a mutz, essentially on top of the kidney. A mutz is a Dutch word for the little white hats that the Dutch kids wear. It's called a mutz. So it looks like a mutz on top of the kidney. If you section through it, as you can see on the outside, you have tissue that looks pretty glandular, and it's organized into epithelial layers, columns of epithelial cells. When you get to the inside, now it starts to look more neural. And these two tissues actually are derived from two different tissues embryologically. This tissue here, the cortex, is derived from your intermediate mesoderm. It also gives rise to the kidney. Your adrenal medulla is derived from neural crest cells that migrate from, they delaminate from the developing neural tube, which will become the spinal cord, and they move into the developing adrenal gland, and they become the chromaffin cells, essentially, of the adrenal gland. Notice that the adrenal gland is divided into 3 layers. They're called the zona fasciculata, the zona glomerulosa, the zona fasciculata, and the zona reticularis. These 3 layers are going to make different types of hormones. So these are stimulated by ACTH, adrenocorticotropic hormone, which is released by the anterior pituitary. And using cholesterol as its biosynthetic precursor, the outermost zona glomerulosa is going to make mineralocorticoids, in particular aldosterone. The first thing you make from cholesterol is that molecule pregnenolone. Pregnenolone is your first precursor. Then look at that, you make progesterone. And progesterone is a precursor both for the synthesis of mineralocorticoids and for the synthesis of glucocorticoids. Glucocorticoids may be made predominantly by the zona fasciculata. That's the middle layer. It's also the thickest layer. Innermost layer, which is pretty thin, is going to take these precursors here, and they're going to make sex steroids, in particular, dehydroepiandrosterone and androstenedione. If you oversecrete ACTH, or if you have a tumor of the adrenal cortex and you overproduce glucocorticoids, you can have a condition called Cushing syndrome. Cushing syndrome. With Cushing syndrome, you will tend to deposit fat in strange places. You'll get what's called a moon face, with this very large double chin. You'll tend also to get tissue deposited back here, and you'll get what's called a buffalo hump. You'll start to get stretch marks in the skin. Skin will start to get thin and lose its integrity because of steroids. You'll have muscular weakness, decreased immune response, tend to get sick easily, hypertension, and also usually early onset diabetes, type 2 diabetes. That's if you have too much glucocorticoids. If you have inadequate production of hormones from the adrenal cortex, you'll have a condition called Addison's disease. If any of you read biographies of John Fitzgerald Kennedy, one of our presidents, the one before Richard Nixon, who was assassinated, he had Addison's disease, although he hid it remarkably well from the public, and he had a very sympathetic press who helped him hide it. But there is a more recent biography of JFK that had access to his medical record. Most of the time, he was a wreck, a physical wreck, even though the press portrayed him as being, because he was young, he was good looking, as being this model of health, essentially. He was physically a wreck, frankly, most of the time. So you have hypoglycemia, and you have ion electrolyte imbalances, you have rapid weight loss, generalized weakness. You can treat the patient, however, with oral corticosteroids, and they can live a relatively normal life. That's what JFK did, essentially. One of these hormones, cortisol, is also known as a stress hormone, and it is secreted during the fight or flight response along with epinephrine, norepinephrine from the adrenal medulla. The ratio of epinephrine to norepinephrine is about 4:1, respectively. Epinephrine also tends to have a higher affinity for the adrenergic receptors in the nervous system and the tissues. So the adrenal medulla is innervated by only the sympathetic nervous system. And so with that mass activation you get with the sympathetic nervous system, you will release these hormones in addition to activating the sympathetic nervous system in general. And other types of stressors can activate the adrenal medulla and the adrenal cortex, cause a release of cortisol. Let me give you an example. We had a Bible quiz this Saturday. Bethany can probably relate to this. Rookie quizzer, she started to turn red, started to forget stuff, make stupid mistakes, pre-jump on the second word. So I called a timeout and I took my quizzer and I looked in. I said, Miriam got up and she walked out. I said, Miriam, and this girl is smart. She's a good quizzer. I said, Miriam, calm down. Calm down. You're turning red. You're getting more frustrated. You're quizzing worse and more frustrated. Just chill. Chill, babe. Chill. She said, I know. I get ready to. And then her mom walked up, which of course was perfect. That calls about God's timing. Mom gave her a little peck on the cheek, said, here, honey, have a drink. Have a drink. Something. It was non-alcoholic, don't worry. It was soda. And she went in and she had 0 with 4 questions left. She got all 4 questions and won that part, got into the finals, and then quizzed out first in the finals and got first place. Okay. That happened to you on an exam? You get this brain lock all of a sudden. All of a sudden, you start to feel a little wet right about here, and sweat starts to go down. And the more stress you get, the worse your memory gets. That's because the blood is going elsewhere instead of your brain. Okay. When you walk in to take a test, you should chill. You should chill. That'll help you remember better. Because these stressors are meant to help you run away, not to help you quiz better or whatever. Of course, I don't think it ever happened to Bethany. She's always just very calm. I don't know, a couple of quiz practices there, darling. You gave me reason for pause. Okay, so that's part of the fight or flight response, which you are well aware of. We already talked about G proteins and the way they do business, what have you. Any questions about the hormones, the endocrine glands we've looked at so far? All right, we are out of time. So we'll stop right there. We'll pick this up and finish it on Wednesday. Don't forget to come ready to drink blue cola and to either walk or run or veg out. Sorry? Meeting in the same classroom. I would advise you, if you have breakfast, make sure it's light. Have a light breakfast, like a bagel or something. Don't like eat 3 waffles and hash browns, and you drink the cola. But if something like that will make you sick, then have something light, a power bar, something like that, what have you. Okay, Brecken, I'll see you tomorrow.